good morning, everyone. I'm Gardner Lapham, and I'm part of the PAME leadership team. And like many of you on this call, I lost a child to SUDEP, and I still search for answers today, which is why I'm especially excited for what we will hear. So I want to welcome you all to this webinar titled Key Learnings from the Center for SUDEP Research. This is the fourth webinar hosted by PAME this year, or Partners Against Mortality and Epilepsy. And for those of you who may be new to PAME, it was started in 2012 as a conference that brought together different stakeholders to increase understanding of epilepsy-related mortality and to champion its prevention. This partnership grew out of an urgent need to respond to the evidence that people with epilepsy have up to 3 times higher risk of premature death than the general population. Today, PAME is looking to expand its reach and increase educational offerings and opportunities to convene various partners to drive change. So I hope you will all join us at the PAME conference on December 2nd in Chicago, or tune in virtually. And you can find more information about that on our website, which is pameonline.org. But now I'd love to introduce you to our moderator, who probably needs no introduction, but whom we are so grateful to have with us today. Dr. Daniel Lowenstein, an internationally known epileptologist, is Executive Vice Chancellor and Provost at UCSF. He has served as Director of the UCSF Epilepsy Center and President of the American Epilepsy Society. He joined UCSF after serving as Dean of Medical Education at Harvard Medical School, where he earned his medical degree. Dan, thank you so much for agreeing to moderate this session, and I'm going to turn it over to you now.
Thanks so much, Gardner, and welcome everyone. And we're just going to launch right into this program because it is filled with useful information. And we also want to have something of a dialogue towards the end so that you can ask your questions. So just an overview, the Center for SUDEP Research was from NIH, from NINDS's most significant investment to date, focused on advancing understanding of sudden unexpected death in epilepsy, or SUDEP. And it's been a large 5-year multidisciplinary and multi-center study that promised to catalyze research on SUDEP and dramatically enhance our understanding of what has been a poorly understood and, needless to say, devastating phenomenon. So today we're going to hear from the program director at NIH, who oversaw the CSR, as well as the lead project investigators. And we'll learn what was achieved. And as I said, we'll have about 15 minutes, we hope, at the end for questions and answers. So I'm going to introduce all 3 of our speakers and let them go at it. So the first is Vicky Whittemore. She's the program director at the National Institute of Neurologic Disorder and Stroke, or NINDS. And she oversees a grant portfolio that includes basic, translational, and clinical studies on epilepsy. And these include the grants on SUDEP, the genetic epilepsies, and seizure localization studies. We'll then hear from Dr. Jeff Noebels, who is a professor of neurology, neuroscience, and molecular and human genetics at the Baylor College of Medicine. He's also director of the Bluebird Circle Developmental Neurogenetics Laboratory, and he serves as a co-PI for the Center for SUDEP Research. And then finally, Dr. Sam Lhatoo is a professor and executive vice chair with the Department of Neurology at the McGovern Medical School. Dr. Lhatoo also serves as director of the Texas Comprehensive Epilepsy Program and co-director of the Texas Institute of Restorative Neurotechnologies. He's co-PI as well for the Center for SUDEP Research. So with that, I'll ask Dr. Whittemore, please, to start off the program. And Vicky, you're muted.
There we go. Sorry. So welcome, everyone. And let me see if I can go back. How do I go back here? There we go. So Gardner gave you a little bit of this timeline, but in 2002, Jean Donalty lost her son to SUDEP, and really then teamed up with CURE to raise funds for SUDEP research in memory of Christopher. In 2004, CURE launched the first grants, SUDEP Research Grants Program. And in 2005, CURE and AES partnered to hold a workshop on SUDEP at the annual AES meeting. In 2008, there was a workshop on SUDEP. I joined NINDS in 2011, and at that time, NINDS and NIH was funding one grant on SUDEP, which was a K grant, a career grant to Alicia Goldman. And I'll come back to the funding situation in a minute. So in 2012, as Gardner said, PAME was initiated. Sorry about my dog in the background. In 2014, the Center for SUDEP Research was funded by NINDS. So from that one grant in 2011, this shows you the funding for SUDEP research over the last 5 years. So it's gone from around 5 million, almost 5 million, up to as much as 8 million. And the estimate for 2021, these are unofficial numbers, is that it's at about $9 million, which is really quite interesting because that no longer includes the funding for the center. So that means that there are a lot of new grants that NIH is supporting on SUDEP. And I also know that there are 3 additional SUDEP grants that are waiting to be awarded from our September council meeting. So there's still great interest, obviously, in the research community and support for SUDEP research at NIH. So how did we get to the Center for SUDEP Research? Well, from those meetings, it became clear that we needed to bring together groups of investigators to really tackle this problem, rather than, and bring them together in a multidisciplinary way. So NINDS first put out a call for planning grants, or the P20 grants, and those were awarded to both Sam and Jeff, who put together groups to put together really very nice proposals that then came in to the request for applications for the Center Without Walls for collaborative research in the epilepsies on SUDEP. We had funding for one center. And so we had the difficult task of bringing Sam and Jeff together with us to try to figure out how we could bring these 2 centers together into one center for SUDEP research that was supported. And you can see here the list of individuals who were part of this project, both on the basic research side with Jeff taking the lead as the PI, and on the clinical research side with Sam Lhatoo taking the lead, who, when he was at Case Western, obviously now he's moved to Texas. But these are the list of investigators plus their teams. This is only sort of the list of the major investigators. There were many students, postdocs, and other individuals who were involved in the research in the center. And all of this research was done and continues to be done in honor and in memory of all those we have lost to SUDEP. And it's our hope that someday we'll understand enough about SUDEP so that we lose no more lives to SUDEP. And with that, I'll turn it over to Jeff.
Okay, thank you. Let's see if I can screen share.
While you're doing that, Jeff, I'm just going to remind everyone to please generate questions, and you can place them in chat or the Q&A, and we'll be coming back to those.
Let's see, there we go. Screen share. Does that work?
Looks good.
Great. Okay, well, I'll begin by thanking the organizers for this incredible opportunity to share a lot of information with you. And before I jump into the scientific data, I need to thank the entire team of people you've just heard about, as well as PAME and NIH for the opportunity they gave us to work together on something that we now sort of refer to as team science, but it was a little bit new at the time. And so what that involved really was a 2-year planning period during which we assembled the expertise and many backgrounds to study SUDEP, formulate the kinds of questions, specific aims, and definitions for each lab. Some of these definitions are kind of existential. I mean, for a scientist looking through a microscope, we have to understand, what is death? When does it begin at the cellular level? And learn how to recognize the events that lead up to and capture the moment of death, because that's in fact what we're trying to understand. And then we spent another year aligning and optimizing the advanced technologies in the pipelines for collecting autopsy specimens, gene sequencing, quantitative MRI imaging, engineered mice and stem cell lines, electrophysiology, monitoring different parameters and brain depolarizations. And then the work began. Let's see if I can advance the slide. For 5 years, we actually went forward and did that, and interacting with each other. And along with the science, we had monthly scientific progress discussions, which were fascinating. We had executive administrative calls to make sure we were all on track. Then we met once a year as a center, and we had other offsite satellite scientific meetings to explore new directions during this period of time. And I guess what it left me with, and I'm still struck by the level of commitment and dedication that we all brought to this endeavor, the exciting discoveries that you'll start to hear about, and finally the optimism that I think we can make a meaningful difference in this very difficult problem. So here's what it looked like. Some of you have seen this slide before, but it's a group of people who are trying to work together to answer some of the simplest detective questions there are, which is who's at risk, when, why, and how can we intervene? And so we decided our goal was to look for clues, which we call biomarkers, both clinical biomarkers as well as basic biomarkers, which would be predictive genes, and better understand the basic mechanisms of why a seizure can be lethal. So you see on the left side, the group that were involved in the basic science work, and I'll refer to them throughout. Let me just grab a pointer. And then just introduce you to the idea of where to begin. So you can either start with the patient, and that involves identifying near SUDEPs or patients that are at severe risk for sudden death, we believe, with techniques that Sam Lhatoo will tell you more about, but also with collecting actual SUDEP cases. That involves scouring the country really for specimens that were taken from forensic anatomies and examinations, and then doing genetics on those. Or you can start from the opposite side, which is start with the genes. So we have indications from mutant mouse models that have epilepsy and have premature death, that there are some single genes that we should be looking at in human. Or we can, even if we're really clever, we can look at gene expression in the brain itself and say, where this gene is expressed might be a very strong candidate for a SUDEP risk. And then, of course, once we have those, we can make models both of human cell types, and you'll hear about the induced pluripotent stem cell analysis. We can look at actual images collected from around the country of SUDEP cases that were MRI'd before death. And then there are laboratory physiology experiments using brain slices and mouse brain to actually see what happens immediately following a seizure. So let me start by giving you a little perspective about 10 years ago, what we thought, what were the mechanisms of SUDEP? And there was very little understood. SUDEP risk was based on population risk. So if you fell into a category of an appropriate age, young age, severe seizure history, if you were pharmacoresistant, that was felt to be a risk for SUDEP. There were no validated biomarkers for specific individual risk. You could not look at a person sitting in front of you in a clinic, epilepsy clinic, and say, I think you are at risk. Because again, so many patients who fit into that population risk category have never had a problem with recovery from a severe seizure. So there also were no recognized brain or cardiac lesions by definition, because we categorize SUDEP as someone who dies for no other obvious clinical reason. So, but people weren't even looking. I mean, they were not doing baseline EKGs routinely for epilepsy patients. They did not scrutinize the MRI and the brainstem, where we now know there might be something to see. So it was the dark ages. And since it was felt at that time that the SUDEP population mortality was about 1 to 1,000, and in fact, people were saying, well, yes, you should inform your patient about SUDEP risk, but you should tell them that that means 1 person in 1,000 might die, but 999 will not. So there wasn't a huge drive to actually warn a patient about this. There was felt to be little to check, just the AED levels and make sure that the person is taking them regularly and doing the best you can to stop seizures. And significantly, there was little evidence for any new biological targets or pathways in the brain where we might learn more in order to intervene in a patient that we defined as being at risk. Now, none of these last 3 are true, and that's because of the work that I'll tell you about based on what the center has accomplished. So some of the major conceptual advances in the last 10 years were driven by CSR labs. Clearly, a lot of other laboratories have contributed to this now, and it's really become a topic for epilepsy researchers. But we feel that we have brought the world of exomes, of imaging and models and stem cells. These are new research biomarkers with real implications for families. So whereas 10 years ago, we thought there was only population-based risk, I couldn't tell you if you in particular were at risk. Now there is solid evidence for at least 25 single genes that confer greater risk than in the population. So you can look at this person and say, well, I'm sorry, we checked and you do have a small risk. Estimating that is another question which we can discuss. There was the idea that pillow suffocation was a leading hypothesis for death. Well, now we've done a lot of work with respiration and in the center, and it turns out that a lot of this apnea is not peripheral smothering, it's failure of central breathing pathways to respond properly at the end of a seizure. And in fact, we have defined some of the pathways from the amygdala to the brainstem that we can look at with MRI. We can test for the reflexes of taking a gasp after a seizure, and we can look at genes that are now known to perhaps depress the reflexes in those pathways. The cardiac contribution was unclear. I remember discussions at the beginning say, well, is it the heart or the lungs or where is the problem? And now we found indeed that there are genes for single genes for long QT arrhythmias and other kinds of cardiac rhythm disturbances that are signaling that this person could be at risk. And we also have developed patient-based functional variant assays to actually not only see that you carry a gene mutation that puts you at risk, but also that your cells are not behaving properly, which is important because not everybody who has one gene is as much risk until you prove it biologically. Finally, the big mystery was it was unclear which seizure will be the last. These patients, we were told, are those who have many severe seizures. So why didn't they have a lethal outcome with their very first one? Something else is happening. And so one of the problems that we've been working on is the hypothesis that a brainstem spreading depolarization, this is a wave of silence that sweeps through a critical region in your brainstem that regulates breathing and heart rate, in fact, is one of the reasons, at least, that would shut things down at the end of a seizure. And we're still waiting for confirmation of this in human, but we have mouse models that show very clearly this is the way it can work. And some of those genes actually are genes for SUDEP risk in humans. So it's very likely that they too have a lower threshold for this silencing wave that I'll tell you more about. I guess the key takeaway is that 10 years ago, we thought SUDEP, well, there's going to be a reason and we'll find out what it is. Now we know that there are actually many reasons and many different mechanisms which complicate matters, but also offer more opportunities to help different kinds of people with different therapies. We'll talk more about that. So let's start with the genes. And this was a key discovery that Dr. Lisa Goldman made. And she found the first gene. It was a cardiac arrhythmia gene. It was well known. But she found that when you mutate this gene in a mouse, and she took 2 different mutations from humans that had long QT syndrome, which is usually announced by having syncopal episodes, fainting spells. But she found that these mice that had these mutations engineered not only had seizures, but SUDEP as well, and in a pattern that is very reminiscent of the ones that have been captured in humans when they've been monitored. A slow, steady decline in the heart rate, and then finally the heart stops after the seizure occurs. And so this was clear evidence that there are genes that can affect both your heart and your brain. And that would be a real simple way of understanding why, in fact, your seizures put you at more risk for death than someone else's seizures. And we traced the expression of that gene. It's in the brainstem. It's in regions of cells that regulate the vagus nerve, that slows the heart. It all fit together. It made a lot of sense in this model that this is how a gene could predispose you to epilepsy. Still, people said at the time, well, that's a mouse, it's not a human. But with some luck, the Mike Ackerman's group at Mayo Clinic, who was doing molecular autopsies of cardiac sudden unexpected death, found that, in fact, when they looked at their marvelous records that they keep at the Mayo Clinic, that up to a half of the patients with cardiac syncope and these known mutations in genes that cause this also were being treated for epilepsy and, in fact, had clear epilepsy phenotypes. So that sort of sealed the deal for us that this was a mechanism to go forward with and find other identifying markers for risk. And in fact, so this is our central hypothesis that there are, in fact, single genes that are linked to SUDEP. Some of them are ion channels. There are other kinds of genes too. They're expressed in both the heart and in the central respiratory pathways. We know less about that, but they are genes, risk genes for post-ictal apnea and lethal cardiac arrhythmias. And as you can see in the image here, that many of the genes, when we look to see which ones are expressed in the heart, we already knew that they were epilepsy genes. We just didn't really put the 2 together. And over on the brainstem side, the less is known about the channels and genes that regulate these different small nuclei in the brainstem that regulate the way you breathe. But we're finding them out, and we have found one, in fact, also already. But so in our laboratory, we've actually studied over 20 of these mouse models. And in fact, not only are there genes that put you at risk, but there are some genes that can cancel that risk. So we're learning more about not only the single gene that we're looking for, but other bystander genes that might be there to help prevent this horrible outcome. So how and where do seizures depress respiration? So George Richardson's laboratory at the University of Iowa has really been a leader on understanding the basics neurobiology of respiratory difficulties, specifically in patients with epilepsy. And his laboratory put out an incredible number, 25 reports on the roles of the amygdala, gene, serotonin, and the carbon dioxide reflex. And so what I'll just tell you in short is that one of the most interesting findings they found by actually stimulating the amygdala of a patient with epilepsy that was having epilepsy surgery. This is a small region in the temporal lobe. And when they stimulated that, it actually would depress breathing. And in fact, the patients were not even aware that they were not breathing. And this is interesting because the amygdala is very commonly involved in temporal lobe epilepsy seizures. So here's a clue of a pathway that could selectively depress brainstem respiratory reflexes. In fact, there are other regions as well. He also looked at some of the specifically genetically identified models in mouse and human, where we know risk is elevated. That's the SCM1A, Dravet syndrome, and SCM8A genes, sodium channel genes. And the timing was important because what he found in studying these patients and mice is that dying begins with respiratory arrest in these models. So it's the breathing that fails first, and that is followed by a cardiac arrest. And these patients have an impaired response to carbon dioxide. So what you do is sort of a brown paper bag test, not quite, but you can test the reflexes of somebody to see at what point they will gasp and recover normal breathing after having a CO2 inhalation. So he also looked at serotonin neurons in the raphe. They did papers showing that these, in fact, are the ones that can depress the recovery from CO2-induced changes after a seizure. And finally, in a subset of patients in the epilepsy monitoring unit, they indeed have blunted interictal bedside reflexes, the hypocapnic ventilatory response. So they can actually test for this while the patient is being monitored for epilepsy. And that's correlated with prolonged postictal respiratory depression. So solid evidence, I think, of a molecular and cellular pathway. So when combined with other risk factors, we now feel that the apnea risk can help stratify individual risks. So we can start to get a little clue as to whether that gene you're carrying really makes a difference in you or not. Next, I want to move on to the imaging. So we had a morphometrics core that was led by Dr. Elisa Goldman and Susanna Muller in San Francisco. And what she brought to the equation was a strong experience and expertise in high resolution imaging and mapping of volumes in the brain. She had been working with Alzheimer's groups. And what she did, together they collected 75 actual SUDEP cases that had MRIs and had to deal with issues that MRIs coming from different centers, all slightly different, but they ironed out those differences and made a very precise and meticulous measurements throughout the brain. And one of the first things they found that Susanna Muller had brought forward were two cases where patients who had died of SUDEP were identified to have a tiny region of atrophy in the dorsal medulla. That's in the brainstem, breathing, and cardiac center. And there was sort of a small hole there. And that drew everyone's attention immediately to the fact that we should be looking carefully at this region in all patients, not just at the forebrain where we know the epilepsy lies. So in subsequent studies, they found that there's really a brainstem network disruption. They've looked at 26 cases with one or more MRIs that have been performed before SUDEP occurred, and they found focal brainstem defects in the brainstem that mediate these abnormal reflexes. So they made two major findings. One is that focal epilepsy is progressive. It can lead to progressive worsening of brainstem pathways. And the second one came out in another study, which is that this damage extends even more widely to involve not only the brainstem, but other regions, what we'll call cardiac central autonomic regions of the brain, other higher centers that send their fibers down to control breathing and heart rate. And so they found evidence of more widespread atrophy throughout this network. And this, of course, takes time to analyze, but it can be done. And in fact, we'll probably learn how to do it much faster so that it can be done on a routine basis. Next comes, and I love this slide because Jack Parent and Lori Eisen were really the first to bring us into a new era of using human actual cells from patients with epilepsy and get induced pluripotent stem cells that you can turn into neurons or cardiac cells and actually judge whether they're working properly or not. And so this is the new era of, I think, personalized functional assays for SUDEP variant risk. We're now doing this for many different kinds of genetic epilepsies, but we did this early on for patients specifically with regard to SUDEP outcome. And so what they can do is make the patient's own brain cells and study them electrophysiologically. They can look at the cardiomyocytes in culture and watch how they beat, either regularly or irregularly, and identify and validate whether the mutation that we think is causative does in fact cause differences in the function of these cells. So what they, in various very nice publications, they identified that there was hyperexcitability and different arrhythmias in cardiac myocytes from this one mouse model in particular, the SCN8A gene. They showed persistent sodium currents when they looked in other cells in the brain. They showed what's called a rapid generation of a virtual patient. So they made these iPSC models for a variety of gene that were implicated in SUDEP, and those are available, I think, for them and other laboratories to look at. And finally, they showed in Dravet syndrome that these patient-derived myocytes show an arrhythmia biomarker in vitro. So you can really say this biomarker, if we see a cardiac arrhythmia, it's because of this mutation, not some other reason. And then there were the idea of bringing this forward to actually demonstrate specific defects to screen for precision therapies in the future. And obviously, we'll not be doing this in every patient, but there are patients that fall into different categories, those with particular sodium channel mutations, et cetera, who maybe once we put them into a group, we'll come up with an answer that will collectively help many patients with mutations in that gene. So let me go back to genetics in a different way, which is to say, yes, there are single genes, but that doesn't mean you're born with only one of them. Elisa Goldman found when she looked at SUDEP in a 3-year-old program and looked at very carefully, she found not just a single gene that we thought put that patient at risk, but 4 genes that we thought put them at risk. So a sodium channel, a potassium channel, a ryanodine receptor channel, and a respiratory control serotonin receptor channel. All of these have been implicated in various ways, but it suddenly realized that you can have more than one reason and therefore more than one pathway. This complicates matters because we might need more than one therapy. But nevertheless, it was a sobering reminder that you have to look carefully at the entire profile and not just stop when you find one single clue. So we followed up on the HT2RC gene because this serotonin receptor, as I told you, is widely implicated in the control of respiration, and there wasn't a clear genetic model for that pathway yet. And so Corey Massey in our laboratory, as well as Elisa Goldman, helped us analyze this gene. And we found that, in fact, when you mutate this gene, you not only get the SUDEP phenotype, but an interesting one. And this tells us how variable SUDEP can be, that this particular gene had a stronger expression in males than in females. And it was a late onset of generalized tonic-clonic seizures rather than a younger age. So for a mouse, this meant it didn't start to seize until it was about 6 months old, which is an older mouse. But we localized the gene. We found it in the amygdala in particular. I can point to that right here. And the seizures that suddenly stop and die. And so this was an important new step forward for pursuing the respiratory defects. And one thing that Elisa was able to do that was really exciting was go back into her SUDEP registry, those 75 patients, and identify 5 cases who were actually carrying mutations in this specific serotonin receptor. And as you can see in that red box, most of them are older than we usually think of. And so there seems to be some coherence with the mouse model and the human model. Interesting, if you look below that, there are 5 cases of SIDS, and those are different mutations, and they all, by definition, occur within the first couple of months of life. But again, raising an interesting discussion about similarities that we can talk about someday.
Hey, Jeff, I'm really sorry to interrupt, but we'd like you to try to close up so we can get on to Sam.
I'm sorry. Yeah,
I got the question. Yeah, the question. I know the questions are pouring in. So
okay. The point of this slide is that maybe we should be doing genotyping in the adult clinic too, because some of these people are carrying risk factors. Here's the last important point, and then I'll lead right into Sam's talk, and that is this wave, this mysterious wave of silencing that goes on in a mouse model. We saw that you can have seizure, and you recover, but then the third seizure, suddenly you don't recover, you die. And in fact, the brainstem undergoes a severe depolarization, the heart stops, breathing stops. And this is what it looks like. We're able to image this in an in vitro slice. And when you give this slice, make it hypoxic, not enough oxygen, you can see that black invasion of that wave, that is actually the spontaneous spreading to polarization that silences all of those critical pathways. And those pathways lead from the lungs and heart and throat and go into the cardiorespiratory nuclei and reticular neurons that activate the cortex. And in the last slide, I just want to point out that when you create one of those waves in a mouse brainstem and you record the EEG, the EEG flattens. Even if the mouse doesn't die, it goes flat. And so that is exactly a phenomenon that Sam Lhatoo, who is a world expert on this, will begin to describe to you about how this post-ictal flattening of the EEG may be, in fact, something that we need to know more about to identify patients. So I will stop right there.
Thanks, Jeff, and we'll pass it on to Sam.
Yeah. Could you unshare? Jeff?
Yeah. Sorry. Stop sharing.
Okay. Can you folks see my slides?
Go to presentation mode. Yeah.
There you go.
Okay. So, thank you, Jeff. And I just want to thank Gardner and the PAME leaders for inviting us to speak today and to share our findings with you, the epilepsy community. Our key areas on the clinical side really were to characterize breathing and cardiac dysfunction in epileptic seizures. We also wanted to find out, following on from some of the work that Jeff mentioned, what parts of the brain were responsible for producing dysfunction, and of course, what parts of the brain were responsible for normal function. In so doing, we wanted to identify clinical biomarkers and imaging biomarkers of SUDEP risk. And we also undertook to create a clinical core, a large prospective multimodality data resource, which we could open up to the research community for SUDEP research. And of course, part of our remit also was to mentor a new generation of SUDEP researchers. So we took a page out of the SIDS playbook. We kind of hypothesized that the critical period, which really is the seizure, along with intrinsic risk factors, the genes that Jeff spoke of, and perhaps extrinsic factors came together in a seizure, in a perfect storm, to create all the bad things that we see in some patients after a seizure. So apnea or cessation of breathing, asystole or cessation of heart rate, reduced oxygenation, increased carbon dioxide, and so forth, all underpinned by neurotransmitters in the brain and brainstem. And of course, you heard Jeff speak about serotonin, and serotonin was one of the main neurotransmitters and one of the main neurotransmitter systems that we focused on the clinical side. Of course, a lot of very clever people had already described a bunch of proposed biomarkers, both for SUDEP as well as for seizure severity. But the interplay between these, what are SUDEP biomarkers, what are markers of generalized tonic-clonic seizure severity, and the known risk factors, which are tonic-clonic seizure frequency, duration of epilepsy, and age of onset. What the interplay was between these, where they intersect, these are of course unknowns, and that's one of the issues that we hope to make inroads into with our work. So one of the first things that we did was to study breathing dysfunction during seizures, and this was work done by Nuria Laqui. You can see a breathing trace in a patient here. This is normal breathing. You can see a seizure discharge and breathing's unaffected, as opposed to this particular patient where you can see that breathing stops when the seizure starts. So there are patients in whom breathing is affected, and there are patients in whom breathing is not affected. And we found that in the majority of patients, so if we look at the duration of stoppage of breathing and if we look at oxygenation, we find that the majority of patients fall into this particular group where the duration of apnea or breathing cessation is brief and the drop in oxygenation is mild. So you can see that oxygenation stays above 90%. But there is this particular group of patients where the drop in oxygenation is severe and where the duration of breathing cessation is beyond 60 seconds. And from observing patients who nearly died of SUDEP in the monitoring unit, we came to understand that this is a potential biomarker for SUDEP, and we should look for such breathing dysfunction in our patients when we see them in the monitoring unit. We also looked at breathing problems after seizures. So this is work done by Laura Vileja. And in this, you can see a seizure ending on the EEG, which is followed by this odd combination of bradycardia or reduced heart rate, asystole or stoppage of heart rate, accompanied by irregular breathing and even stoppage of breathing. So this is a patient who nearly died of SUDEP, and we came to find that in approximately 20% of our patients, some of whom later on went to have SUDEP, that this is a problem that occurs and again serves as a potential biomarker for SUDEP. We looked at serotonin levels. This was work done by Arun Murugesan, who at that time was a medical student, along with his mother, who actually happens to be our senior research associate. They looked at serotonin levels in these patients and found that serotonin levels go up after a seizure. Interestingly, in patients who have breathing difficulties, that rise in serotonin after a seizure is not as high. So there's an interesting interplay here, as you would predict from what we know about serotonin and its role in breathing, that serotonin levels have an influence on breathing problems after a seizure. So Jeff already spoke about these studies, but what I want to point out here is that our focus during the course of our investigations in the CSR increasingly came to be upon the brainstem. So the hypercapnic ventilator response, and this is work done by Roop Sainju, Brian Gelbach, and George Richardson in Iowa, is where increasing carbon dioxide in the air that a patient breathes is accompanied by an attempt by the body to get rid of that excess carbon dioxide. So that results in an increased rate of breathing, or so-called minute ventilation. So this is what you would expect: increased CO2, increased breathing. Here is a patient who did not do that and later on went to develop or die of near SUDEP. Now, this is a brainstem reflex. And as Jeff mentioned earlier on, his work with SUDEP mouse models showed this very interesting and lethal phenomenon of spreading depolarization followed by spreading depression that results in the brainstem shutting down and causing the animal's death. So the brainstem really is where final control of blood pressure, heart rate, and breathing, all these things reside. So we understood from Suzanne Mueller's work, again, as Jeff mentioned, that the brainstem indeed has a problem in patients with epilepsy. And very interestingly, we also came to understand from Suzanne's work that this increase in atrophy and the risk of death happened closer to death. So there was a temporal or a dynamic aspect to this observation. So Maria Thom, who was the neuropathologist in the CSR, studied a bunch of SUDEP brains. And this is an important study, very systematically done, in that she looked at Dravet brains. She looked at the brains of patients with epilepsy who did not die of SUDEP. And she also looked at control patients who did not have epilepsy, but had died of sudden death. And she made several very important observations. One was that the serotonergic neuronal system in the parts of the brain in the brainstem, the median raphe and the ventrolateral medulla, these are areas that are involved in breathing control. there was a depletion of neuronal populations in these areas. And she also made the observation that the longer the duration of epilepsy, the greater the damage to these brainstems. So we are now increasingly focused on the brainstem as an area that is important in the pathogenesis of death. So we went a step further. So we took Roop Sainju, George Richardson's work, and Suzanne Mueller's observations. And what we did was we gave carbon dioxide to patients while they were in the functional MRI scanner. So you can see here that the oxygenation remains level at about 100%. We give them increasing CO2 here and here. And what we see is very interesting. So when we compare 10 people with epilepsy and do a grouped analysis against 10 healthy controls, you can see that the parts of the brain, so this is the brainstem again, the parts of the brain that regulate breathing just light up in these patients who have epilepsy. This indicates a brainstem under stress in dealing with carbon dioxide, and this raises the possibility that we are nearing the identification of imaging biomarkers that can be used pre-mortem in patients who have epilepsy. One of the contributions of the CSR is making cardiorespiratory monitoring standard in many epilepsy monitoring units. So here we look at blood pressure, breathing belts, carbon dioxide, oxygenation, and so forth. And so we were able to identify other types of cardiorespiratory dysfunction. So here you can see a patient in the green who has postictal hypotension. So this is a group of patients who have postictal hypotension. And we now understand from analyzing this data that there are problems with cardiac output in these patients in the post-seizure phase and in a decrease in total peripheral resistance in these patients. So you can imagine that in patients who are not breathing well and their hearts aren't pumping very well, that a drop in blood pressure is going to be catastrophic. So to understand where or what parts of the brain were driving function and dysfunction, this is work done again by Nuria Laqui, where we mimic seizures by passing current through the brain. And we find that these areas highlighted in red, amygdala that Jeff spoke about, initial work done by Brian DeLuhy and George Richardson in Iowa, were indeed the places where if the seizure spread would result in cessation of breathing. So here you can see electrical stimulation of the amygdala. You can see breathing here. And during the stimulation period, the person stops breathing. Similarly, this part of the brain, so-called Brodmann area 25, we found to be responsible for blood pressure. So here is the stimulation of Brodmann area 25, and you can see the blood pressure dropping in this patient, and it takes a while to recover.
Hey, Sam, again, I'm sorry to interrupt, and we're leaving you even less time, but there's so many questions coming in that I think we should make sure that we get into the conversation mode.
Yes.
So if you can.
So I'll finish with this slide then. The question is, identification of these brain sites for dysfunction is fine, but are there therapeutic applications to this? And here you can see that low frequency stimulation of the amygdala with specialized electrodes results in an enhancement of breathing. And this work is now funded by Cure and sets the stage for breathing rescue in patients who have trouble after seizures. So I should also mention that the work done in the CSR now has resulted in this multimodality database where we have 2,700 patients with a variety of data sets. We have DNA material in these folks, EMU materials, MRI, and this is an open resource for research. I just want to mention these folks who are currently using the CSR database for their research. 3 of them are funded by Cure. Lisong here is funded by NINDS with an R01. And my thanks to all these centers that provided data for these data resources. I'll stop here. Thank you.
Thanks so much, Sam. And Sam, sorry to cut you off much earlier as well, but again, there are just so many good questions coming in. And just one general comment, as someone who's been in the world of epilepsy and epilepsy research for many decades, like some of our colleagues, I'm overwhelmed by the progress that's been made. It's astonishing how much more we know now than 10 years ago. And these investigators and their teams and NINDS with Vicky's leadership deserve a huge, huge amount of credit for moving the science forward. And of course, PAME and Gardner and the parents and the families have been the motivation that's made all this possible. So this is remarkable. So a whole bunch of questions. Let's see how many we can get to. Will any of these findings change existing guidelines and how providers counsel patients? Sam, you want to start with that? And then Jeff?
Yes, that's a great question. And as I mentioned earlier on, I think one of the contributions of the clinical collaborations in the CSR has been in making cardiorespiratory monitoring in the epilepsy monitoring units a standard. And I think we now know enough to create guidelines and to encourage the use of both cardiac, that's EKG, multi-channel EKG, and respiratory recordings in the epilepsy monitoring unit, because you discover things about tendency to breathing dysfunction, all the kind of things, the bad things that can happen that can put a patient's life in danger. So these things, I think, should be encouraged and should be done, and will certainly change the way we look at risk in our patients.
Jeff, a couple questions, of course, related to the genes. One would be, what do you think the current approach should be for genetic testing, especially in folks who are concerned about the risk for SUDEP? And related to that, has anyone examined epigenetic regulation of chronic seizures on SUDEP genes in the brainstem and other nuclei of interest?
So that's two great questions, because first of all, we're being promised that someday sequencing will be free, in which case we'll sequence all of our epilepsy patients. But the fact is, it's still a complex matter to interpret the sequence data that you get, as Dan Lowenstein well knows. And the idea that you would counsel somebody based on a cursory finding of, oh, I see, there was a loaded gun in your DNA, but there were no bullets inside, because there are other genes that could protect against that. So you have to be very cautious, particularly with this diagnosis, of saying, okay, you are at risk. And that's why we've proceeded very slowly to validate the different kinds of mutations in that gene that might be risky. The second thing that I know from looking at all of these genes in mouse models is that they don't all die. Most of the genes cause death in maybe half of the colony. The other half do not. So that's by definition some kind of epigenetic influence on whether this gene becomes a major and prominent defect or not. That could be even non-genetic. It could be environmental. So on the other hand, some genes are very powerful, and they certainly give you 100% predictability. So I would say the easiest question is, it all depends. That's always the answer. But I think we can move into a new zone where, first of all, we develop checklists for practitioners to say, would I even want to get a sequence? Would I do a clinical exome study in this patient? So we have to get them thinking about looking for SUDEP risk and spending the money, time and money and effort to understand it. And then, of course, always is, what would we do with this information? Do we have anything yet to intervene? And without that, it's difficult to make a strong argument, unless it's your child, to find out the risk. But I would say even then, do you really want that concern when you don't know how valid it is? And so this has been the dilemma from the beginning, I think.
And epigenetic effects of seizures on the expression in these nuclei that seem to be put people at risk? Any evidence yet of that?
Oh, yeah. No, I think seizures remodel all parts of the brain, and in some people more than others. Diet can change it. The therapy that they're on can change it or prevent certain things from happening. So there are just so many aspects to the plasticity that we need to understand in order to answer the question, why did it happen today and not tomorrow? Mm-hmm. Something led up to this, and we don't know what it is.
And also trying to tie again together a couple of questions related to the changes in the actual structure of the brainstem that's been observed now, which is just a remarkable finding. Do you think that we should now have a special epilepsy brainstem protocol for imaging that becomes standard, the way we have, for example, when someone we suspect temporal lobe epilepsy, we do certain sequences? Should that be part of regular practice now? 1,000%.
Yeah. I think there is no reason, if you're doing an MRI on one of these patients, the way technology is advancing, there's no reason, I think, in the near future that we couldn't automate something. The question is compared to what? And so that's where Susanna's large database may be helpful in identifying for a certain age patient, given the number of seizures they have, is this out of the range? But these things can be solved.
Mm-hmm. Sam, any comment on that one as well?
Yeah, I think as Jeff says, the brainstem is definitely a part of the brain that we need to focus clinical studies on. I think it's a little too early to go to your epileptologist or to your neuroradiologist to ask for comment on the brainstem in standard clinical MRIs. But certainly on the research side, we're working towards making that doable on the clinical side.
And a question that sort of links together the breathing functions and epilepsy, overlaps between SIDS, sleep apnea, and the disturbances that are involved in SUDEP. Any comments on, you've alluded to a little bit of that in terms of the genetic basis with Alicia's work, looking at genes that are common. Anything more to say? If there's a family that in which sleep apnea runs in the family, is that an added concern?
I think the answer is yes, but how much concern depends. It depends on the age. There are many other things that are linked to sleep apnea, and it's often seen in older, overweight individuals and throat issues and collapse of the airways and things like that. But in the SIDS group, that was by far and away the leading category because no one had ever seen anything else going on in these children. They hadn't had a seizure, but that could be because either seizures are subtle or they just haven't had one yet. And it turned out that the apnea overcame them. Apnea can kill, there's no question about it. And Hannah Kinney and others who looked at many SUDEP brain regions saw evidence of the kind of pathology that we see in human temporal lobe epilepsy in those SIDS cases. So there is some biological evidence that these are two sides of the same coin.
And I see we're at the top of the hour. So Gardner, can I turn it back over to you, or do you want to use one more minute? Your call.
You wrap it up, Dan. You've done such a great job. If you want to squeeze something in.
Okay. One more question. People are hungry to know what kind of intervention can we do, here in the fall of 2021. If you're a family member, if you're a parent, what are you recommending?
I think there's a lot of exciting work that's being done. I briefly alluded to neuromodulation that takes advantage of what we know about cardiorespiratory dysfunction. One of the things that I didn't speak about is that we published some data from the CSR on the role of SSRIs and benzodiazepines and their association with cardiorespiratory dysfunction. So there's something to that story. We confirmed some of the findings that previous investigators had reported. And so I think we are now, particularly with the serotonin story, I think we're nearing a phase where we can do a randomized controlled trial of pharmacological interventions for SUDEP.
Well, that's a great place to end, actually, because the involvement of so many of the people who are listening in today is going to be critical for our ability to make progress. I'll just close, Gardner, by saying this. I always wonder about what the historians are going to say about these times. And there is going to be a day where in the book on the history of epilepsy, there's going to be a chapter about SUDEP. And that chapter is going to begin with the complete uncertainty that we fumbled around with for forever until the kind of work that's happening right now. And that chapter is going to end with the end of SUDEP. I don't have any question about that. And it's going to happen sooner than we think. So thank you, everyone, for really a remarkable hour together. And as you can see in the chat, PAME is going to be meeting in Chicago. And we certainly welcome everyone. Gardner, back to you.
Yeah, no, thank you all. We had the A team today, and this was just phenomenal and inspiring and gives us a lot of hope. And I just want us all to work together to leverage what you all have built here, because there are more avenues to keep going down to make this happen and realize Dan's vision and write the end of his chapter of that book. So thank you all.
Thank you.
Great day.
Bye-bye.
Bye-bye.